Forget. I even set my phone to tell me that. Liliana, any ideas there? I'll give you a hint. The enzyme is called isomerase. What's an isomer? What did we do? What did we do to glucose? It changed into what molecule?
I don't know. It looks like they're similar, but
Different shape?
yeah, the shape is different.
That's all it is. We changed the shape, right? Don't get hung up on this stuff, guys. It's easier than it seems. Good job. All right, Perla, you got the next one. What do you think happened in this picture?
More ATP?
Used or gained?
Gained.
How come?
Well,
Where's the arrow going on the ATP?
To the right.
Look on top of the arrow here. Is the ATP being, is it going into ADP or is it becoming ATP?
It's becoming ADP.
ADP. So are we using ATP or are we gaining it?
Losing.
Losing, right? So we invested one here. We invested one here. We've invested two. Who's keeping track of the math? We're not doing so well so far. The whole point of glycolysis is to make ATP. We've already lost two. Good job, Perla. Daniel, what happened in this picture? Again, I just want you guys to see it's not rocket science here, right? Don't memorize these steps.
Split up.
It's split into two.
Mm-hmm.
Nicely done, sir.
Oh, okay.
See what happened there?
That's why we need two ATP.
Well, it's just that's how many it takes to get it to break into half.
Oh, I just thought it was like one for each half.
No, but I like the thought process. All right. Oh, I got you a hard one here, Gael. What's going on in this picture? It continued on from the last slide. What are we making in this slide? Look at on top of the arrow.
I mean, I see that you're using 2 NADH.
Using them or making them? Which way is the arrow going?
So you're making them?
You're making them, right? These are those golden stitches, right? I'm going to put them in my pocket because we're going to send these down the electron transport chain. Gifted student. Ari, you got the next one. Look on the arrow. What are we making in this case? Look at all these intermediates, right? But we're finally doing what, Ari?
You're making two ATPs.
Yeah, we used two, we made two. Who's keeping track? How many ATPs are we up now? Any? Or we just break even?
We broke even.
We broke even. We still haven't done anything yet, right? Oh, you guys are awesome. Good job. All right. I'm out of chart room here. Let me see. Jada, you get the next one, okay? By the way, you guys can just answer them without me calling on you if you want. That's a silly step. You don't have to worry about that one. All right, how about that next step, Jada, over here? What are we making again? Look on top of the arrow. It's going from...
ADP.
What's that?
ADP.
We make an ADP or we make an ATP? Which way is the arrow flowing?
ATP.
We're making ATP. We have a net gain finally of ATP. Now remember, we invested two and now we made two, right? Sorry, we made four because it's split in half. So it's a net gain of two ATPs and we're done because look what we're stuck with, pyruvate. Daniel, you were looking for the formula. There it is. The 3 carbons tells me it's pyruvate. I broke glucose, a 6-carbon base, if you will, into 2 pieces of 3 carbons. Where'd the electrons go? Oh, here they are. They're in NAD. They become NADH. Where's the energy at? Oh, in the ATPs. We're done. Now, if you're a bacteria or you're running a mad sprint, that's all you can do is glycolysis. Glycolysis does not take oxygen. Glycolysis does not involve the mitochondria. It's outside of the mitochondria in the area we call the cytoplasm, or sometimes you'll hear the fancy word cytosol. And the whole point is to break glucose in half to make ATPs. Now, I got a little something to tell you. How long do you think a multicellular organism is going to last on just 2 ATPs? Very long? No. Try running a mad sprint for very long. You aren't going to be able to do it. You got about 90 seconds, you're done. Glycolysis is the splitting of sugar. What do you guys think? Questions?
Well, yeah. I came up with one. Like, I see around the end where that 2 ADP just comes out of nowhere. Like, are those just free ADPs, or are they?
Where are these ones that are coming out of nowhere?
Yeah.
Here?
Oh no, around the end.
Which
The
number?
ones we're,
Number 10?
yeah, the ones we're gaining.
Oh, the ones we're gaining aren't coming from anywhere. It's just that my muscle just used them. Know what I mean? Remember now, these are recycled 10 million per second per cell. So every time I use my muscles, I'll get ADP. And every time I break another glucose molecule down, I turn them back into ATP.
So it's like a pit stop.
It's like a pit stop. I like to think of it as like a charged and an uncharged battery. The ADP being an uncharged battery, ATP being charged. Now, if you don't believe me, try this experiment at home. Go home, sit in a corner, don't eat for a week, and see how many ATPs you make without ingesting glucose. The answer is none. Huh? I'll cut that out.
Okay.
Yeah, I learned that yesterday. I learned how to edit. I was very proud of myself because, you know, somebody told me not to stop the recording, so it went for an hour and a half. So I got crap. Now I got to figure out how to edit. Oh, your old teacher is learning. Not very fast. Yeah.
I have a question. You said that whenever we move our muscles, we break glucose bonds, and then that
Well,
helps.
careful. When you move your muscles, we don't use our paycheck. We don't use glucose. We use the cash. What's the cash of the cell?
ATP.
ATP. So if I want to move my bicep, I put a phosphate on my bicep muscle. That's what makes it move. And then I got to keep. Go ahead.
How do we turn ADP to ATP though?
That's in the mitochondria. That's what we did right here. When we broke glucose down, right? The end result is ATP. I cashed my paycheck, glucose, into ATP. By breaking glucose, I'm making ATP.
Okay, thank you.
Is that for sure? That's got to make sense, that part. All these little steps don't matter, but that's important. Good? All right, those are the steps we went through. Now, here's how it was on the AP exam day one day. They really made it shorter. And again, notice they're never going to ask you, hey, tell me all the steps of glycolysis. But they might expect you to understand what's going on in the steps. So if you look at the top, for example, we're breaking glucose apart. Oh, sorry, not yet. We're not breaking glucose apart, but we're investing 2 ATPs. So it might say on the test, where either A, B, C, D, or E are we investing ATPs? Well, it's here at A, isn't it? How do I know I'm using ATPs and not gaining ATPs? Well, it's a simple fact of looking at the arrow, right? I'm making ADP, not ATP. That's the energy investment phase. What do you think? I pause for effect.
Good.
Mm-hmm.
Good.
Good?
Yes.
I'm not that good of a teacher, guys. Somebody's got to have a question.
I mean, yeah. I see on A and C, phosphates are being added to whatever's going on. But in A is when we're investing, but C, the phosphates come out of
Yeah, I'll get the C. That's a tricky one, buddy. Hold C.
Yeah.
Hold that thought 1 second. Now in B, they'll ask you on the test, they'll say, hey, where does fructose break into 2? And you might say, oh, I don't know. Well, look right here. You put a 2 in front of this molecule, there's 2 of these PGALs. The name doesn't matter, but it's phosphoglycerolaldehyde, right? Who cares what the name is? But there's 2 of them. See that? Now, Daniel, you bring up a good point. Here, I'm literally adding inorganic phosphates. They were floating around in my cytoplasm. That throws a lot of people. This reaction is so exergonic that it is possible to just add phosphates, right? If we're breaking that vase, so much energy is coming out of this that I can add just a couple of phosphates to this molecule. But they call them inorganic because it's not coming from ATP. But let's look at letter D, especially if somebody's not sure if they know what they're doing. What happened in letter D here? What did we finally do?
Turned it into ATP.
We finally made ATPs. We lost 2 up here, remember? We made 2 here, so we're at even. It's not until step E that we actually make a net gain of ATP. In glycolysis, how many ATPs we make? A total of 2. We had to invest 2. We made 4. 4 minus 2 is 2. That's all we made. If you're a bacteria, that's all we're going to be able to do. If you're running a mad sprint down the hallway, all you can do is glycolysis. And 2 billion years ago, when we were not multicellular eukaryotic organisms, this was all we could do. When we swallowed a mitochondria, right? When you were 1 cell big anyway, when you swallowed a mitochondria, boy, now we can make a lot more ATPs. That's the citric acid cycle, and that's what I want to show you today if we get that far. Questions on it? You guys are doing fantastic.
Yeah. Just one question.
Yeah.
You know how all animals go through this, right?
Correct.
So like they make glycolysis. So
And plants, don't forget. Plants do cellular respiration.
oh yeah. I'm just having this question, like what happens to the ATP or the ADP or phosphates or whatever that we eat from animals? Like, do we absorb it?
Yeah, we can use those too. Yeah, we can use those too. You know, all the nutrients we take in, the phosphorus for ATP, yeah, that came from animals originally, sure.
The mitochondria?
Yep.
Oh, so we're literally stealing mitochondria?
Yeah. Kind of borrowing them though. Remember, they break apart so fast and reform so fast. You know what I mean?
Mm-hmm.
But yeah, you're using their parts.
Mr. Charline, so then all of that process that we just went through happens in the mitochondria, right?
Careful now. The glycolysis does not take place in the mitochondria. Everything else I'm about to show you does.
Yeah, it's the thing like just before, right? Like in the picture you showed us.
Yeah,
where,
which picture you want? All the way back here?
no, I know which one. Yeah, the one where it just shows like the mitochondria and then the cytosol.
Yeah, that's what I thought you were talking. Hold on, I'll get there. Not that one. It's coming. There it is, this one. So here's glycolysis. We made ATPs. We made a couple of NADHs, but we're not in the mitochondria. Today, I want to go in the mitochondria in this area called the matrix, and we're going to do the citric acid cycle. Now, if you're catching on, right, and most students around the country are not going to catch on to this, they're thinking, oh my goodness, I got to memorize all these steps. No. What would be the point of the citric acid cycle? What do we got to do to pyruvate still? Think big picture now. What do we got? Yeah, we still got to steal electrons, right? We're going to steal every electron from glucose. Remember, it started as glucose until it comes out as what? Think about the reactants of cellular respiration. What's a naked glucose molecule going to look like? take a breath out and tell me what's coming out.
Carbon dioxide.
Carbon dioxide. That's when we'll know we're done. If we steal all the electrons from glucose, it should come out as carbon dioxide. Good?
Mhm.
So we just did glycolysis. We're going to take those 2 3-carbon sugars, pyruvate. We agreed there's still electrons to steal. And now we're going to go into the citric acid cycle. Now, the fact of the matter is, we can only do this if there's oxygen. If you're running a mad sprint down the hallway, you don't have enough oxygen to do this. So you will only do glycolysis. And as Daniel pointed out yesterday, we produce this byproduct called lactic acid, makes our muscles tired, right? But let's say we're just here listening to a lecture, and now we're going to take pyruvate, we're going to combine it with oxygen, and we're going to enter the citric acid cycle. Now, Denise, we do need a mitochondria. All right? All good?
Yeah.
All right, so we went through those steps of glycolysis. We're not going to memorize them, right? We're going to realize we had to invest a couple of ATPs to get this whole thing started. We had to put a little energy in, but we gained a net gain of 2, which frankly isn't much, right? When we're done, we're going to get 36 ATPs. So the next step is the oxidation of pyruvate into this enzyme called acetyl coenzyme A. Acetyl coenzyme A is just a 2-carbon sugar instead of a 3-carbon sugar. So we're going to take a 3-carbon sugar and knock off our first carbon dioxide. And what was the point of that? What are we going to steal? What are we stealing from these molecules?
Electron.
Electrons. We're going to oxidize this molecule called pyruvate. It's going to turn it into a 2-carbon sugar called acetyl coenzyme A, and we're going to waste our first CO2. Right? It's going to be, you're going to breathe it out. If you just joined us, we're on page 3 in the middle there. We're about to go to the citric acid cycle, which, remember, I'm an old teacher, so if I keep calling it the Krebs cycle, they're one and the same, but we should call it citric acid. Again, don't memorize these steps. So here's what happens. Here's pyruvate, a 3-carbon sugar. By the way, I put this PowerPoint on Google Classroom, so you can just look, right? You can go back and look at these, right? I'm going to enter the mitochondria using one of those transport proteins, and there's the release of my first CO2. Hey, I'm getting close to done, ain't I? If I'm making CO2s, we're almost done. That's a naked glucose molecule. What I'm going to be left with is a 2-carbon sugar. Now, question. Do you think a 2-carbon sugar, is there still electrons to steal? What do you think?
2
Yeah.
carbon.
Yeah, there's definitely. We'll know when we're done, when all the electrons are stolen, is it's going to come out as CO2. And look who got the electrons. Can anybody tell me that?
NAD.
NAD became NADH. NADH has lots of energy. It's like that golden stitch. I stick it in my pocket and it's buzzing like my cell phone. That reminds me when I got my cell phone for the first time, I thought I stepped on like an electrical wire. I thought I electrocuted myself. I didn't know there was a buzz feature. Like, oh no, I think I'm killing myself here on some electrical wire. I know Daniela, or not Daniela, Rebecca, you just think that's funny. But she told me to record, so that's good. So here it is, citric acid cycle. How are we going to know when we're done with the citric acid cycle? All of our glucose is coming out as CO2. When we've oxidized glucose, in other words, when we steal the electrons from glucose, we'll know we're done because CO2 is going to be left. Now, I'm on the page, if you want to write these in, I'm on the top of page 4, and I'm going to come back to the picture on page 3. But anyway, it says on page 4, if you want to write those in, the cycle oxidizes organic fuel derived from pyruvate, and we're going to generate 1 ATP, 3 NADHs, and 1 FADH2. Now, we talked about the difference between memorizing and internalizing, right? Somebody tell me, what do you think FADH2 must be? By the way, before it was FAD, and now it's FADH2. Here's my perfect point of not memorizing all biology. If we know how biology works, then we can figure these things out. What do you think happened if it went from FAD to FADH2? What did it get?
An ATP?
You got what?
An ATP?
ATP? Where would you see that in here? How would that work? You're on the right track, though. What's the energy source? What are we trying to steal?
The electrons?
Electrons. So what do you think FAD, when it becomes FADH2, what do you think it got?
Electrons?
It got electrons, right? Just like NAD became NADH, it got electrons. So who knows why there's two different molecules here, but there are two different molecules. Their job is to take the electrons originally from glucose, and now these things have more energy, right? FAD has less energy than FADH2, just like NAD has less energy than NADH. These are the guys with the electrons, and you still don't know what we're going to do with them, right? That's the last step, what we're going to do with those electrons.
Mr. Treveline, I have a question.
Yes, please.
Why is it that the H represents like the energy? Is there a reason behind it? Or
Yeah,
it's just...
what we're doing now is stripping those electrons from molecules, and so what you're left with a lot of times are just protons. So these H's, I know that's weird for everybody, but can we just pretend like when it gets that H, that's what's giving it the electrons? When you see the H on the molecule, I just want you to think, oh, it got its electrons. And it stole those electrons from glucose. It's a really, really good question.
Wait, Mr. T,
Yeah.
did you say that NADH has less energy now?
If I did, that was a mistake. NADH has more energy than NAD.
Okay.
Did I speak?
I think so.
Yeah. Catch me on those, please.
Wait, what was that?
I said that NAD had more energy than NADH. That's not true. NADH has more energy than NAD.
Oh, okay, thank you.
Yeah. Everybody okay? So if you don't mind, I'm gonna put up the whole citric acid cycle. I'm going back to page 3 on the bottom. Some people like to write them all in. Remember, you're gonna have the PowerPoint. There's no need to write them all in. And the reason there's no reason to write them all in is because you're gonna have this exact thing on your AP exam, right? It'll tell you everything you need to know. But what's the point of the citric acid cycle? Think about that just for a minute, please, and tell me what you think the point of the citric acid cycle is. Take a look at the products, and what are we making? Now remember, we're stealing electrons from glucose, which is now pyruvate. Pyruvate is going to go around this complex citric acid cycle. Oh, look, I see CO2s. See them? It means I'm almost done. So who got the electrons now?
The oxygen?
No, not oxygen.
FAD.
Yeah, FAD became FADH2. It's got the electrons. And who else got electrons? One more.
NAD.
And NAD became NADH. And now we're done. We stole all the electrons from glucose. How do you know we stole all the electrons from glucose?
You create ATP.
We did create ATPs. But how do you know we stole all the electrons from glucose? What's it coming out as now? Think of the big equation.
CO2.
CO2, right? So we are done stealing all the electrons. And we haven't made all our ATPs yet. Citric acid, the point of this cycle is to take the electrons originally from glucose, give them to NAD so it becomes NADH. You guys can see my little arrow here, right, when I'm doing this?
Yeah.
And it's also going to make electrons from FAD become FADH2. Oh, and look, we made a couple of ATPs on the bottom here as well. On the very bottom of this page. So you say, Ms. T, what would they ask me on the test about this? Well, they'd give you the whole cycle and they would say, what's the point? And you're going to tell them the point is to take the electrons from those molecules, all those intermediates. By the way, here's why they call it the citric acid cycle, because the first intermediate is citrate. Sorry, I gotta help Daniela got kicked off. Right? So the point of this is to make NADHs, FADH2s, and ATPs. Now, if you're thoroughly confused, that's good because we don't know what to do with all those NADHs and FADH2s. But remember, they're the ones with the energy. They're the ones that have the energy source. Go ahead.
So in other words, eat your oranges.
Oh, yeah, I guess so, yeah. For the citric acid. Yeah. Well, you got to eat all these things, right? You talk about consuming animals. It's oxaloacetate, isocitrate, all of these things, they are consumed every day, right? They're part of your nutritional value, yes. Now, the interesting thing about this cycle is you end up at the exact same spot that you started. As long as you continue to feed acetyl coenzyme A into this cycle, you can keep this thing going. But the cool part about this is the product, the last step here, is the beginning of the next step. As long as you keep eating glucose, you'll continue to make NADHs, you'll continue to make FADH2s, and you'll continue to make some ATPs. But the fact of the matter is, we still have not made enough ATPs for somebody like us to run around and do all the things we want to do. We need to make at least 36 ATPs per glucose molecule. And if you want to count them up, there's not many on there.
Mr. Traveline, so just to clarify, what is the purpose of the intermediates in the cycle?
Okay, I'm going to give you the philosophical answer to that. Those intermediates, we're trying to break the vase a little at a time. If was that Denise that asked that?
Yeah.
Yeah, Denise, if I gave the electrons right to oxygen, you haven't even seen oxygen yet, like my car does, I would waste so much to heat that we wouldn't be able to survive. The point of all those intermediates, the point of all those little steps is we're trying to break glucose a little at a time. By having it fall a little steps and not one big step, I'm going to be much more efficient than my car. My car, about 25% efficient. You, 40% efficient.
Okay. So they basically help you get as much out of
we're correct. We're trying to lose less to entropy.
Okay. Thank you.
Hold on, Mr. T.
Yep.
I'm just looking at the bottom, and I came up with this question like, does it not like directly give a phosphate to ADP? Because it looks like something.
It does. These ATPs down here and the few that we made in glycolysis, we're going to call those substrate level phosphorylation, and it's almost a shortcut. This is a very exergonic reaction, and so there's enough energy to make a few ATPs almost by accident, if you will, Daniel. And those are made by what they call substrate level phosphorylation. That won't make total sense until I show you the electron transport chain. The electron transport chain is where we're going to make most of our ATPs.
Yeah, it's because it looks where it looks like something called GDP takes the phosphate and gives it to ADP.
That is correct. Now, GTP, I'm not too worried about yet, but if adenine is the nucleotide, G is another nucleotide in DNA called guanine. And eventually I'm going to say, hey, you didn't need to know that. If this is adenosine triphosphate, this must be guanosine triphosphate. Again, if we know a little biology, we can apply it to other things. If you try to memorize all this stuff, you're in a lot of trouble. Right? So we're internalizing. Yeah, and would you have realized that thing had 3 phosphates, Daniel?
Yeah, it's
Or at least
TP.
what's that? Yeah, it's 3
I know.
phosphates, triphosphate.
Mhm.
So who cares what the letter is before that? But our main ingredient is this. And the reason we care about this is because this is what makes my muscles move. This is what makes the sperm cell move. Remember, we did the little tail dance, right? Every time I got to add ATP. This is the source of our energy for the most part in our body.
So GDP is just the messenger.
Yeah, intermediate, if you want to use that
Okay.
word again. And somebody way smarter than I am figured out all these steps. In fact, it was Krebs. That's who came up with this. They named it after him. And then later on, I don't know why, but they changed it to citric acid cycle. On your test, it'll say citric acid. Nobody calls it Krebs anymore, and I'm not quite sure why. All right, how much time I got left? All right, we're going to write less, listen more at this point, because I want you to see the end. I want you to see what we're doing. So far, you're like, yeah, great, Mr. T. What's the point of all this? Well, the point is we oxidized glucose. We stole the electrons from glucose. We know that there's no electrons left because they're coming out as CO2, a byproduct, a waste product. We breathe it out every day, right? That's the main gas coming out of your mouth every day, right? And I still haven't made a lot of ATPs. So there's a third step, and that third step is called the electron transport chain. It's a nice name for it because what are we doing? Well, we're going to take all the electrons that we got from NADH and FADH2, and we're going to send them down the chain. Now, I'm going to draw the chain almost like a staircase. And the reason I'm drawing it as a staircase is because I want you to realize that we're letting it fall one step at a time. Who can tell me why we don't want to go right to oxygen? What would happen if we send all those electrons right to oxygen? Anybody? We'd lose most of our energy to what?
Entropy.
To entropy, right? That's exactly right. So again, we're going to see 1 million intermediates. We're going to see 1 million steps. Don't let that throw you. Remember, the point is to make ATPs. Now, with the 5 minutes left, I'm going to talk fast and show you the whole thing. I don't expect we'll pick it up again next time, right? Where we left off. But the source of the electrons now are NADH and FADH2. Let's back up just a minute. Who had the electrons originally was glucose. Who has the electrons now is NADH and FADH2. Who's going to get the electrons ultimately? Well, take a deep breath and tell me who's going to get the electrons. I'll strangle you if you don't get this right on the test.
Oxygen.
Oxygen. That was not a threat. That was a hint, right? It's the oxygen that's going to get the electrons. When the electrons get to oxygen, we are done with this entire mess. And it's just a series of oxidation, reduction, oxidation, reduction, oxidation, reduction. Take the electrons from one molecule, you give them to another. So this is it. This is the last step. Look at down here, and here's oxygen. I told you a long time ago that oxygen is very electronegative. It loves electrons. So that's why it's here at the bottom of the chain. As long as you continue breathing in oxygen every day, it'll be at the bottom of the chain. Now, here is FADH2, and here's NADH way at the top. My question to you is, which one do you think is going to make more ATPs? NADH here at the top, or FADH2, which is lower on the chain? Which one would you think would produce more ATPs in the end?
NADH.
Which one?
NAD.
NAD, why?
I'm looking at the existence free energy relative to O2, so it seems like it has more free energy.
Yeah, it's starting further up the stairs, isn't it? Here, if I take the electrons way up here, they fall, they fall, they fall, they fall. Every time they fall, I'm releasing energy. FADH2 enters the system much lower on the staircase, if you will, huh? And so it only falls a few intermediates instead of all these intermediates. The fact of the matter is, you can make up to 3 ATPs with 1 NADH. Its weaker cousin, FADH2, only about 2, because it falls less steps. Now the electrons are going to fall down this chain one at a time. We're going to take that energy, and we're going to take the electrons, and it's going to go to oxygen. There's the other byproduct of cellular respiration is water at the bottom. But you're still looking at me going, Mr. T, that's great, but I thought we were going to make ATPs, right? Here's where it gets crazy. And once you get this, you won't figure it out today, but once you understand this, once you comprehend this and let this wrap around your head, you're going to realize that we did all this 3 days of lecture just to do one thing. And let me show you what that is. We're going to pump protons out of the mitochondria into this, what they call this intermembrane space, and we're going to let them flow back in through this molecule called ATP synthase. ATP synthase is one of those transport proteins. Look at protein, phosphate, lipid, lipid, phosphate, protein. And it turns kind of like a turnstile. We're going to let those protons flow through that ATP synthase, and look at what we finally make. ATPs.
Yeah.
I'm going to get out of here. This class must start. But let me keep talking as I'm walking. I promise I'm almost done. So again, maybe tonight at 3 a.m. it'll dawn on you. We spent 3 days of chemical reactions for one very dumb reason: to pump protons out of a space just so that they can flow back in, so that they can physically turn ATP synthase.
Wait, Mr. Troutline, where did the protons come from?
Great question. Remember all those electrons we stole?
Mm-hmm.
Now look what it's left. You steal electrons, you're left with protons.
Mm-hmm.
See that little H plus?
Mm-hmm.
That's it. Now, I've got a question for you. How come those protons have to go through ATP synthase? They're awful small.
I guess because they have a charge.
Because they have a charge. Remember the two things that have a hard time getting through the membranes? Large and charged. So we're going to use these little charged particles. There's no way they're going through these phospholipid layers. They're going to make them pay the price, and they're going to go through these transport proteins. Now, if you were in class, we would all line up, and I'd make you stand in the corner. I usually do this every year. And you guys would be the protons. And then you'd want to go sit down, and I'd make you flow through me. And on one of these little spinning chairs, oh, I have a big coat and everything. It's crazy. We'll do it sometime when we're here. And I make you guys go through me. And every time you go through me, you turn the turnstile. And if you turn the turnstile, guess what we make? ATP. Lastly, what do you think this transport protein is called? It's got a great name. It's called ATP synthase, because it makes ATP. Remember, the point of cellular respiration was to make ATPs. We are now done. 3 days of lecture. We're almost done. We'll finish it up tomorrow. But 3 days of lecture just to pump these protons out so that they diffuse back in through the transport protein called ATP synthase. Why so many steps? Because we don't want to lose it all to heat. This makes us about 40% efficient. Compared to your car, about 25% efficient. Think about that next time gas prices go up. Hey, extra credit. Maybe you can do it tonight. Come up with an ATP car. What do you think? I'm only asking for 50% of the profits. All right. Bye, Ms. T. Have a good day. You guys were awesome today. Great job.
See you.
I'm gonna press record or stop record.